Welcome back. Today's topic is about the somaclonal and the gametoclonal variations. So now question is why the variation happens, right? So generally we know that the plants undergo a series of cell division, particularly the mitosis cell division, and the cell division is very rapid and uncontrolled. So because of this uncontrolled division, it leads to the variation, and it is of three types. First is the somaclonal variation, then the gametoclonal variation, then the protoclonal variation. So we have discussed in the tissue culture the plants that derive from the tissue culture have been variously that referred to as the somaclones, and the variations displayed by such plant. So that somaclones they will display some kind of variation. So that variation caused by the somaclones are the somaclonal variation. In the same way, the chelicones are the clones of callus, the protoclones are the clones of protoplast, and the mericlones are the clones of meristem. Right? Then about the somaclonal variations are observed among plants regenerated from callus cultures of somatic explants in the in vitro culture. Then the gametoclonal variations are variations shown by plants which are regenerated by the gametic culture techniques like anther culture or ovule culture. We'll discuss in the list class about the anther culture. The next is about the protoclonal variation can be seen in plants regenerated from the protoplast derived callus cultures. Okay. So, according to the Lerkin, in 1986, somaclonal variation is the genetic variability which is generated during tissue culture, right? And the variations for karyotype, the isozyme characteristics, and the morphological variations in somaclones have been commonly observed. Okay, and such variations themselves act as a heritable mutation and persist in the plant population even after transplantation to the field. Okay, so then what are the sources of the somaclonal variation? So the sources it could be the epigenetic factor, the genetic factor, sometimes the physiological factor and the biochemical factor, but now we are discussing about only the epigenetic factor, that is first the characteristics is leads to methylation, then DNA amplification, activation of the transposable elements. Somaclonal variations occur spontaneously in low frequency. So if it is the epigenetic change, then it will be in the low frequency, and the epigenetic variations are very temporary, just like the phenotypic changes, and it is reversible. Then about the genetic cause, in case of genetic cause, obviously the change will be in the DNA. So there will be change in the chromosome number, okay, that leads to euploidy, that is the change in chromosome set, and aneuploidy, that is change in parts of chromosome set, and then polyploidy, that organisms with more than two chromosome sets, and monoploidy, organism with one chromosome set, right? Then this is about the chromosome number change. Then there is chromosome structure change, okay, and that leads to the deletion, inversion, duplication, and translocation, okay. And the next thing is uh, the frequency is very high in case of genetic cause, and changes in the cytoplasm genome is also a cause, and it's are very stable and irreversible. Okay. Then next is about I told you there are factors that are the biochemical. The cause is the lack of photosynthetic ability due to the alternation in the carbon metabolism, then the nitrosyl metabolism, then the antibiotic resistance, then biosynthesis of the starch via carotenoid pathway. I have not mentioned it here, the biochemical cause. And next will be what are the advantages of the somaclonal variation. So the somaclonal variation, induction of the somaclonal variation is very cheap. It is a very cheap process and quick method for inducing the variation. Then next is somaclonal variation can be induced through the application of pathotoxins, herbicide, salts, metabolic inhibitions or temperature shocks. Then it helps in the crop improvement. Okay, as it induces the mutation, right? Then we can detect the environmental stress, the which species is resistant to herbicide and which is disease resistant and which is environmental stress tolerant. Correct. So it helps in the crop improvement. It gives the genetic variation, that is the mutation. Then it is very suitable for the breeding of tree species. And some of the disadvantages are it requires extensive and extended field trials. We need so many field trials for this. Then it is not suitable for complex agronomic traits like yield and quality. And it may develop variants with pleiotropic effects which are not true. So next is the steps involved in the induction and the selection of the somaclonal variation. So first, from a single cell there is the culture, the callus tissue formed. Then there is the process of organogenesis. What is the meaning of organogenesis? In case of organogenesis from a callus tissue, as I said, this is a callus tissue. From the callus tissue, the root, shoot, and the other organs give rise. And in the next step, after the organogenesis, you can see the regenerative plants. Then there is the hardening and the selling process, selfing process, and the somaclonal variants will get. Okay, so the organogenesis first, if you will take the high oxygen to cytokine ratio, then we'll get the roots, and if we'll take high cytokine to oxygen ratio, then we'll get the shoot first. Okay, so this is all about somaclonal variation. Here is one question. The question is somaclonal variation is by the which culture? Callus culture, anther culture, meristem culture, or the cell culture? So the answer will be callus culture. Once the callus will produce, then the callus tissue will give rise to the other organs, that is the root stem and the other organs by the organogenesis process, right? So by the callus formation, the other organs are produced. So the method of organogenesis through the callus formation relates to the regenerative plants, and then the hardening and the selfing process, and we'll get the somaclonal variant. Okay, so this is all about the somaclonal variations. What are the causes of the somaclonal variations? What are the advantages? And what is the difference of somaclonal variation and the gametoclonal variation? What are somaclones, caliclones, protoclones, and the mericlones? So in the next class, we are going to discuss about the anther culture. If you like this video, hit the like button, share with your friends, and do not forget to subscribe this channel and press the bell icon so that you will not miss a video from this channel. Thank you very much.